Hello and welcome to today's PIR live event brought to you by Partners in Research Canada. My name is Stacey Joyce and I will be your host. Remember that you can ask questions throughout the webinar by finding the chat button in the top left-hand corner of your screen if you're joining us live, and make sure you select all panelists and attendees in the to field so that all of our audience members can see what questions are coming in from across the country. If you include your name, your school, or your city, we'd be happy to give you a shout out when we ask your question. Today's guest is Dr. Brad Doble. He is an associate professor in the Department of Biochemistry and Biomedical Sciences, and he is a principal investigator at the Stem Cell and Cancer Research Institute, all at McMaster University. Thank you so much for joining us today. I will let you take it away.
Thank you very much. It's my pleasure to be here. Can you see the first slide there, Stacey?
I can indeed.
Okay. So I'm one of 8 investigators at the Stem Cell and Cancer Research Institute that's located in Hamilton. And we all work on various aspects of stem cell biology. And my focus is on something called stem cell signaling, with a focus on what kind of maintains stem cells in a stem cell state and what allows them to change into different types of cells. And we'll see that these sorts of processes have to occur properly, and if they don't, that can lead to some diseases such as cancer. So I'm studying signaling for stem cell biology purposes, but also to understand what happens in cancer. So what is a stem cell? So stem cells are a very powerful type of cell. They're a unique cell in our body. They're the cells that give us the ability to regenerate when we have injuries, and that their properties are such that they can make copies of themselves in a process called self-renewal. So that self-renewal can be where they make exact copies of themselves. So that's called symmetric division, where you have 2 mirror images, basically, of the original cell. But they can also do something really important that most other cells cannot do, which is to make a perfect copy of themselves, but also give rise to a cell that's not the same. So that's called an asymmetric division, where we have a copy and a new type of cell. And that new type of cell is called a progenitor. So those progenitors can also make copies of themselves, and those cells are the ones that will give rise to the specialized tissues that we have in our bodies, such as, for instance, neurons. So here's a couple of neurons that came from these, what would be neural progenitors. And this whole process is used throughout development, and it's also what is used to keep our bodies nice and fresh. We have all sorts of tissues that kind of break down with time, such as the linings of our intestines. And those cells that have to be replaced are replaced by stem cells. So there's different types of stem cells that have what are called different potencies. So there's one type of stem cell that's called a totipotent stem cell. So these are the rarest, and I'm not really going to focus on them much. But that type of stem cell would be able to give rise to all of the cells that we have in our body, as well as extra embryonic tissues, such as the placenta. So the cells that I will be talking a lot about are called pluripotent stem cells. So these are very similar to the totipotent stem cells, with the only difference being that they can't make extra embryonic tissues. So anything that's not part of the whole body, such as the placenta, cannot be made by these stem cells. But everything else, so an entire, say, human or mouse, which I study a lot, can be made just from these cells alone. Then we have another type of stem cell called a multipotent stem cell. And these are a bit more restricted in their ability to make different types of cells. So a good example of this would be a blood stem cell, which can give rise to all the various types of blood that we have in our blood system, such as the red blood cells, the white blood cells, and the various different types of white blood cells that we have. And then kind of lowest on the totem pole are the unipotent stem cells. Those can only make one type of tissue, and one of those cells would be the skin stem cells. So one of the stem cells that you may have heard a lot about are called embryonic stem cells, or ES cells. So those are a type of pluripotent stem cell. And I just wanted to give you an idea of where these stem cells come from, because when people talk about embryos, they usually have in mind something that maybe looks like a small human or a small mouse. But the embryos that stem cells come from, embryonic stem cells come from, I'll show you. They look not like a mouse or a human in any way. So right after fertilization, we have a single cell that's formed that's called a zygote. So that is totipotent. It can make everything, including the placenta. Then we go to a 2-cell stage. That's still totipotent. 4-cell stage, 8-cell stage, something called a morula, which is just one stage past this. And then we get to this structure that is still really, really tiny. And it's from this structure called a blastocyst. There's this mass of cells in the blastocyst. That's a really, really early embryo. So this is a mouse blastocyst, and it just looks like a sphere with this blob of cells in it. It's these cells here that give rise to the embryonic stem cells. So the embryo is tiny. So this is kind of roughly the size. This is the tip of a pin. So the type of embryos that are being used to generate embryonic stem cells are really, really, really early embryos. And that's why these cells have the ability, because they're so early in development, we can actually make an entire organism from those cells. So in my lab, we use these cells in experiments where we can mix those embryonic stem cells with another kind of host embryo, and from that we can generate entire mice. So I'm just going to show you one example. This is not from my lab, but our lab does similar things, where they used embryonic stem cells that have a green fluorescent protein in them. So this green fluorescent protein is originally from a jellyfish, but it allows us to see these cells using fluorescence microscopy. And by taking those cells and putting them into another host embryo, we can get these mice that are running around that are also fluorescent, because those mice are made from embryonic stem cells. All of the cells of these living green mice came from the original green stem cells. So that's kind of one of the really powerful things that stem cells can be used for in experiments, is to generate new models that can be used for studying diseases. So I just want to give you an example, kind of an analogy for how cells function. So all of us nowadays use computers or iPads or iPhones. And for these computers to work, in the background, there's some sort of computer code running. So there's a program that is required for your computer to do what it does. And that code, a lot of times you might have heard of computer bugs or glitches. We have the ability to edit that code. So if there's mistakes, we can go in and we can make changes to the code and make it better. And we'll see that in a way, cells are quite similar to little computers. So cells, in the kind of center of a cell, there's something called the nucleus. And in the nucleus, we have information. So it's similar to the information that you would have in a computer. In a computer, that information is stored as zeros and ones. In the cell, in the nucleus, it's stored in this molecule called DNA with different types of molecules. So there's 4 different types of molecules that form the code in our body. And every single cell of our body that has this nucleus has all of the information to make an entire human being. So it's quite incredible. There's a lot of information in that one little nucleus. But that information has to be interpreted and executed throughout development, but also throughout our entire lives. And that whole process of kind of getting the cell to take that information and use that information to allow it to function, we're still figuring out how that all works. And that's one of the things that my lab is really interested in. So cells have these programs that let them become kind of what cells they are. So a neuron, so the cells that are in our brain and our spinal cord, they have neuron-specific programs in their DNA. Muscles have muscle-specific programs in their DNA. So they all have the same DNA, but the programs that are executed are different for these different cells. So the pluripotent stem cells that I talked about also have their own unique programs. And in these cells in particular, we have 3 proteins that are really, really important for executing the program. They're called OCT4, SOX2, and NANOG. And they form a kind of a circuit in these cells, talking to each other and regulating the cell. And this is just human pluripotent stem cells. These green things are the nuclei that are stained for these different factors. And you can see that all the nuclei are just loaded with these different factors, showing how important those factors are for these particular cells. Other cells don't have these factors. They're unique for these pluripotent cells. So one really incredible thing that was just discovered not that long ago, in 2006, is that we can take any cell of our body and using just 4 of those factors, including the OCT4, the SOX2, KLF4, and c-MYC, so those are just factors, you don't have to remember the names, but we can take 4 of these factors and integrate them into our DNA using viruses, and there's approaches where you don't even have to integrate them, but they can change any of our cells into one of those pluripotent stem cells. So this is a remarkable thing, which a lot of people wouldn't have thought is possible. But it means that you can take blood cells, skin cells, so these cells here were skin cells, and make them into pluripotent stem cells that you can then change into whatever cell type you want. So it gives us a way of studying things that we couldn't study before in the human. So somebody that has, for instance, a brain disorder, we can't take a brain out of a person and study it while they're alive, but we can take some of their blood and make it into neurons, and then we can study those neurons and see if we might learn something about their brain disease by studying the neurons that we generated using this approach. So that was only in 2006, and the importance of this is reflected in the fact that he, in 2012, only 6 years later, got a Nobel Prize for that. So there have actually been many, many incredible things happening in the world of stem cell biology and just biology in the last few years. So I mentioned that you can quite easily edit computer code. So very recently, it has become quite easy for us to edit the programs that are in cells as well. So this is something that my lab has been using for a few years now as well, to try to understand how cells work and how cancer cells work. So this particular technique is called the CRISPR-Cas system. You might have heard about it. It involves small molecules attached to a protein, and we can make these quite easily in the lab. The important thing is that these specific molecules can recognize very specific regions of the genome. So we can target these molecules to specific regions of the genome. And when that enzyme, that's the blue thing here, is recruited to a region of the genome that we recruit it to, it causes a double-strand break in the DNA. So that information is cut. And normally what will happen is that you'll get a repair happening that isn't quite right. So it actually, you will usually disrupt that region of the genome. So that can actually be a quite powerful way of getting rid of the function of a particular gene to study its function. But another thing, a very powerful thing that we can do, is we can supply a template molecule as well. So we can make the cut, so to make that cut that we've made in a very specific place, then add a template, and we can change that piece of DNA, so we can change that code that's in the cells to whatever we want it to be. So if we have a diseased cell, we can correct that diseased cell genotype, if it's a genetic disease, by using these approaches. So this is something that is very new and very powerful, and will really change a lot of biology that we'll see very soon. So as far as what we kind of can currently use stem cells for, there's something called regenerative medicine. So we know that some types of organisms, such as the newt here, you can cut off an entire limb from a newt, and in just 6 to 9 weeks, the entire limb, like the whole leg, will grow back. So there are people out there that wonder and study how this process occurs. So why can a newt do this and a human can't? Or can we? Like, do humans have this ability? To a certain extent, yes, we do. So if you cut your finger off and you don't go beyond the bottom of your fingernail here, your finger actually has the capacity to grow back. Especially the younger you are, the better this will work. But we do have some limited regeneration on our own, and this regeneration properties use stem cells. And there's a lot of people around the world that are trying to understand how this process works so that we might be able to grow back other damaged parts of our body when they become damaged. So another way that we can use stem cells, and this is currently being used in Ontario, at least I know, just recently within the last few years, is the use of stem cells that come from the eye. So there's a region here around the eye, just between the white of the eye and the cornea. There are stem cells called limbal stem cells that can be harvested even from people that have damaged eyes. So these people here had acid splashed into their eyes by accident. And these eyes with the acid splash damage, they cannot see through these eyes at all. But they still have some regions of stem cells from which they could harvest some stem cells and grow fresh new corneas outside of the body and then transplant them. And we can see that this can fix this problem perfectly. So this is 6.5 years after this treatment was used, and this has taken somebody from being blind to being able to see. So the last thing I'm going to kind of mention is that, so that code that I talked about, the information that's stored in our cells, in the DNA, so there's all sorts of signals that come in from the outside of the cell that alter what happens with that information. So that's why our different cells behave differently and why our different cells look differently. It's because they only use different regions of that code for their functions. And those are regulated by a bunch of different, some have some pretty strange names like Wnt and hedgehog, but different types of signaling pathways feed into the information that's kept in the DNA. And we need these things to work properly for our cells to be normal. So what happens if we have problems with those pathways? So if we have mutations in the genes that are part of these pathways, you can have massive changes to what normally would happen. So these arrows here are depicting that there's some information coming out of this code, and it's a specific amount of information or a specific amount of, say, cell division that has to be really tightly regulated. And if it's not tightly regulated, if we have mistakes happening anywhere in these pathways or mistakes in the code itself, you can get this high level changes that make things really, really different than they were. And this, when you have a few of these things happening, they add up on top of each other and can lead to cancer. And cancer and stem cells might sound like they're different things, but cancers basically are very similar to stem cells in that they have this self-renewal ability, except it's not controlled. So normally our stem cells, I said that you can have that asymmetric division where you have the stem cell split and make a new cell, and that's very, very controlled. If it's not controlled, you can get expansion of those cells to such a high extent that they cause tumors to form. And those tumors to form can then spread to other parts of your body, and they basically kind of take over your body. So the worst kind of cancers they'll call undifferentiated cancers, meaning that they don't really look like any of the other cells that we have in our body. They really look like stem cells. So there's a really close link between stem cell biology and cancer cell biology. So that's why in my lab, we're studying cancer stem cells to understand kind of how they normally work. We're using this CRISPR technology to manipulate the genome, to study specific regions of the genome and specific genes, and trying to get an idea of how we might be able to treat some of these cancers. So kind of in a nutshell, that's what my lab is doing, and the kind of research that we're doing right now. And I'm going to stop there, and I'll take any questions that you might have.
Well, thank you so much. If you want to go ahead and click the stop share button, we'll be able to see you a little larger. And I think you already explained one of our questions, which was what is happening inside the body when someone has cancer. So that sort of unchecked cell division, that more and more division happens until it results in a tumor. But why do people lose weight when they have cancer? That question is coming from a grade 10 student at Subway Academy 1.
Okay, so it kind of depends to a certain extent on what type of cancer it is and how far the cancer has progressed. But what happens is that, so your body, so a normal, say, teenager should eat maybe 2,500 or 3,000 calories a day, and that's enough calories to keep all of the tissues of your body happy, so enough nutrients to feed all of the cells that you have in your body. What happens when you have cancer? So cancers, because they're dividing so quickly, they basically eat the food that you would normally eat, especially if it's so that the cells of your body need those nutrients. But when you have cancers that have a very high metabolism and are getting larger and larger, and a lot of times they're actually going through a process where, you know, they're getting larger and then some of them are dying, but then you have new ones replacing the dead ones. So it's kind of a perpetual, very highly metabolic state where these cells are basically using the nutrients that the normal cells of your body would normally be using for themselves. And there's other, you know, depending on what kind of cancer it is. So if you have a leukemia, so that's a cancer of the blood system, basically what happens there is normally you need all the different cell types that you have in your blood system. They serve different functions. Some of them are for your immune system. Some of them are for carrying oxygen to your tissues. What happens in the cancer scenario is that you get something that's called a leukemic blast cell. So these cells basically crowd out all of the other normal cells, and those cells only have very limited normal function. So it's kind of like crowding your blood system with cells that can't really do anything. So you go from having a blood system where you have all of your nice functional cells that are working properly to having a blood system filled with all of these more or less useless cells. So that can also be very damaging and can lead to weight loss, because, you know, if you don't have your red blood cells getting the oxygen to your body, you try to make up for that by eating, but you'll just kind of waste away. So it's basically the tumor is taking the nutrients that you would normally be getting.
Well, thank you. I think next we'll go to a question from Joyce in Mississauga, Ontario. Can you be born with a tumor? Is that possible?
Yes, you can be born with a tumor. So tumors can either be cancerous or benign. So benign means not cancerous. It means that you can just cut it off and it won't grow back. So those are just kind of abnormal growths. And it can be possible for some babies to be born with tumors. A lot of times those tumors are benign though, and they just have to be removed. Some of those tumors can be called something called a teratoma, which have a bunch of different types of tissues in them. So these can even develop in adult people, usually in the gonads. And what happens is you can get these tumors that will have hair and teeth, and they're very bizarre. But they're, they have all of these bizarre structures in it, but they're highly differentiated. And I mentioned before that it's the bad tumors are the ones that you would call undifferentiated, that look more like a stem cell. So those ones you can just cut off, and you'll be okay for the most part.
I have a bit of a lengthier question here for you, but
Okay.
I think it's quite interesting. From BPCI, if every cell contains the same DNA, but with different programs switched on, if patients are treated with stem cells, can those stem cells develop the same mutation? And if so, is there a way to modify the stem cells to keep them resistant?
Yes. Yes. So I'll tell you about one sort of example that might answer your question. So in the blood system, so those leukemias, there was a girl actually in the United Kingdom who had one of those blood disorders, a type of tumor, and they couldn't. So the way that disease would normally be treated was by giving transfusion. So what they do for a lot of different types of these leukemias is they get rid of your normal stem cells. Or so if you have the leukemia, they might be cancerous. So you'll have these cancerous stem cells and cancerous cells. They will actually treat you with high doses of radiation, so whole body radiation, to kill all of your actively dividing cells. So that includes the cells of your hair and skin and intestinal lining, all of your actively dividing cells. So it's not something, it's not very targeted at all. It's not an awesome treatment, but it also kills the stem cells of your blood system. Then they come in with blood that they get from a donor. So you might have heard of, bone marrow transplants. So they'll collect the bone marrow from a donor and then take that normal bone marrow, so all of the blood cells, including stem cells from that donor, and put it into the recipient. So normally that can treat a lot of different types of blood cancers. Not all of them. There's some that you can't use that approach. But this one girl had a disease that normally could be treated that way, and it didn't work. So they tried all of the conventional treatments that would be used to treat this particular disease. But what they were able to do is take her own stem cells, blood stem cells, modify them using a kind of a CRISPR-based approach. So the cells that she had weren't working properly. They're kind of attacking her own body, I think, was the problem. But they were able to prevent that from occurring by using a gene editing technique. And when they put her own cells that had been corrected back into her, she was able to be cured, or at least she's in remission still. But everything else had failed. So this is where these gene editing techniques are going to be very powerful, because they give us the opportunity to relatively easily correct DNA damage.
So an interesting question on the flip side, very short question from Jay.
Okay.
If you know the cause of cancer, why can't you stop it? Simple question, but I don't think the answer is a simple answer.
No. So it's because we kind of know the cause of some cancers. And so the issue with cancer is that every type of cancer, so even say one type, call it breast cancer. Breast cancer is not one disease, it's hundreds of diseases. And each of those different types of breast cancer has different mutations and different conditions. It's kind of like every human is different. We all have different DNA. Cancers are also very different. And we now know that even within one tumor of one person, different regions of the tumor can have different mutations. And kind of the set of mutations that each of those pieces has can work together to give the cancer. So it's not as easy as kind of you mess up one gene and you end up getting cancer. It's usually many genes are mutated that work together to lead to the cancer. So it becomes a very complicated issue to correct.
I see. So next, I'm going to go to a question from Assumption High School in Burlington, Ontario. They'd like to know your opinion on HeLa cells being used in research.
Okay. So, yeah, that's a good question. So HeLa cells are cells that, here we have them. I think probably every lab that does cell biology work has at least access to HeLa cells. And these were originally, they came from a tumor from one woman, and it was kind of without her consent that these cells were used to generate these lines that have been used for studying cancer for many, many years now. So, so there definitely are ethical issues with this. I think it's just the problem is that ethics kind of are developed as the issues arise. You can't really, so before that, there really weren't any cancer cell lines that people were working with routinely. So just the fact that they were able to get those lines kind of growing was kind of a feat of its own. And you can't kind of set up ethics for something that kind of hasn't even really existed before. So now there are definitely very strong ethics in place for any type of donation of tissues towards any sort of medical research. You have to have informed patient consent, but that's only because we know better now. So those cells have been very useful to the scientific community. We've learned a lot by using those cells. So she didn't give consent, but she's definitely the Henrietta, I believe her name was, has definitely enriched science with her contribution, even though I guess it wasn't by consent. One would hope that she probably would have consented if she knew that she would be, being so beneficial to understanding how cancer works. But yeah, I guess that's my opinion. Is that
it seems very, very complicated, and especially when you dive into the ethics and that things are sort of clearer moving forward, but it sounds like it may never be clear for that particular cell line.
Yeah, for that particular case, it's kind of hard to go back in history and kind of change what happened. But now, so with all of these CRISPRs, it's so easy to manipulate the genome now that there has to be a lot of ethical thought going into, what can we do with these CRISPRs? Because theoretically, we can go into a zygote, human zygote, make any genetic changes we want, so you can really kind of custom make any species now. So the ethics associated with the new technologies that are emerging, we really have to make sure that we get those in place, and so we don't have these similar sort of situations occurring where the ethics kind of lag behind.
That makes sense. But yet, probably easier said than done. So we'll see how things play out.
We'll see how things play out. Yes.
So we've already gone a bit over time here. We had such a great discussion going. I didn't want to interrupt, but we should wrap it up for our classes who are still with us. And I just wanted to give you an opportunity, perhaps, to comment on. I know we had a question from Mr. Fisher's class in Toronto about other potential uses for embryonic stem cells, or I thought you may want to comment on that, or what opportunities are out there for students who may be interested in pursuing research in this area of biology.
I think there are a lot of opportunities, especially in the Toronto area. So I didn't mention it in this talk, but stem cell biology was born in Toronto, really, initially with the hematopoietic, so the blood stem cells. Those were first discovered by Tilla McCullough from Toronto. And there has been a history of stem cell research in Toronto and the GTA kind of ever since. So we've Canada and Toronto area has actually been have been leaders in stem cell research, and I know there's still a lot of active stem cell researchers in that area of the country and throughout Canada. So the opportunities for studying stem cell biology, there are a lot of them, and it's really moving into many different fields now. So regenerative medicine, there's a real push for this now. Now that we're getting better at kind of growing stem cells in culture, the idea is to be able to generate artificial, well, not really artificial, but tissues from the stem cells by using a kind of a combination of engineering, bioengineering, making scaffolds to hold the cells. And Toronto, I know they're actually working on that as well. So it's kind of a combination of engineering and stem cell biology going hand in hand to make new cartilage, new muscle, new skin. So I think there are a lot of opportunities for really cool cutting edge stuff in Canada and definitely worldwide right now. It's still a very big thing, and I can only see with the advances in genome editing together with the stem cell techniques that it's just going to become more and more used.
It will be very interesting to see how things go, even in the next 5 to 10 years. You told us today about quite a lot of advancement in the last 10 years. So I'm sure there'll be so much more in the coming 10 years. But for now, that is all the time we have. We'll check in with you 10 years from now and see what the story is. Thank you so much, Dr. Doble, for joining us today and sharing your expertise.
Oh, yeah, it's my pleasure. Yeah,
and,
thank you.
Next week on PIR Live event, we are talking about renewable energy. More information on this and other live event webinars can be found at pirweb.org. Thank you so much for joining us today, everyone. We'll see you next time.